Parkinson's disease is the second most frequent neurodegenerative disease and affects over 6 million individuals globally. Within the brain's dopaminergic neurons with PD, abnormal aggregation of protein called alpha-synuclein is observed. The aggregated alpha-synuclein is thought to be closely linked with PD pathogenesis. Inhibiting the spread of aggregated alpha-synuclein into neighboring neurons may contribute to reduction of PD pathology. However, two major challenges are faced when treating PD. These are delivering drugs to the brain and targeting abnormal alpha-synuclein aggregates specifically. Brain drug delivery is challenging because of major differences in the blood vessels compared to the rest of the body. This maintains an ideal environment while protecting the brain from harmful foreign substances and hormones and neurotransmitters destined for the rest of the body. The smallest blood vessels in the body are called capillaries, which are made up of endothelial cells. Capillaries are responsible for exchanging nutrients, waste, and other molecules in the blood with tissues throughout the body. This exchange mostly happens through gaps between endothelial cells. Capillaries are also found in the brain. However, their structure is very different. There are no gaps between the endothelial cells. Instead, there are specialized tight junctions. Surrounding these capillaries are pericytes and astrocytes, which regulate exchange and the formation and maintenance of the brain's blood supply. Combined, these components make the blood-brain barrier. Unlike normal capillaries, the BBB restricts access to nutrients, waste, and small lipophilic molecules such as fatty acids. Big molecules like biologic therapeutics, including antibodies, severely struggle to penetrate. Only up to 0.2% of circulating antibodies pass through. To improve BBB penetration rate, ABL BBB bispecific antibodies use a unique best-in-class BBB shuttle technology to pass through to the brain and central nervous system. They contain a BBB receptor targeting region, which is an engineered antibody developed to selectively bind to a BBB receptor. As traditional antibodies do not have this region, they cannot recognize the receptors mostly found on BBB endothelial cells. However, ABL BBB bispecific antibodies can bind. After binding, they are taken into cells and transported across the BBB through a process called receptor-mediated transcytosis. ABL BBB bispecific antibodies are efficiently delivered to the brain and central nervous system because the target receptor is not as abundant anywhere else around the body, making them highly adapted to target brain-related diseases such as PD. As ABL-301 contains a BBB shuttle, it primarily localizes to the brain and passes through the BBB at a much higher rate through the receptor-mediated transcytosis. Once inside the brain, it targets abnormal aggregated synuclein with superior therapeutic efficiency. It was shown that ABL-301 prevented deterioration in movement-related symptoms and reduced aggregated alpha-synuclein inside the brain in the preclinical disease models. Also, side effects are predictable and manageable in preclinical models. ABL BBB bispecific antibody platform can be adapted for the treatment of other neurodegenerative diseases, including Alzheimer's.